Hello there, you're welcome to Biology Lectures with Akirili Oladimiji Philip. It's my wonderful pleasure to have you with me on the lecture. So today move on to cell structure in the Gcse past question solution series. All right, so we're going to start from here. This is the beginning of the question. It says the image below shows an epithelial cell from the lining of the small intestine. So you might want to pause the video here to actually go through all of this. This talks about the direction of in which food is being absorbed. All right, so he says the first question here says, what is the name of the part? A, talking about this structure right here, and that is the nucleus. That's the nucleus. Please do remember to pause the video, attempt the question, then play. All right, moving on. How are most soluble food molecules? This is a key word you don't want to forget. When you say soluble food molecules, absorb into the epithelial cells of the small intestine. Osmosis only deals with water, so osmosis is out. Respiration is about absorption, it is about making of energy. So the answer here is diffusion. Please don't forget why osmosis is wrong, because osmosis talks about how solvent, which is water, move across a partially permeable membrane. So you don't want to forget that part, so to say. All right, moving on. It says suggest how the highly folded cell surface helps the epithelial cells to absorb molecules of food. Well, what I would say is this whole thing you're looking at, really, if it's stretched out, it's going to be like so long, which means it increases surface area of the, let me write it here, increases surface area for the absorption of substances, or if you say food molecule, it's still fine. All right. Now it says here, epithelial cells also carry out active transport. Okay, name one food molecules absorbed into epithelial cells by active transport. Well, you should remind yourselves at this point that what's active transport to start with? That is the movement of molecules or substance from area of lower concentration to area of higher concentration. That's a bit strange, by the use of energy. Now, why I said a bit strange is because normally, imagine you have 1, 2, 3, 4, 5, 6, 7 here, and you have just 1 and 2 here. According to active transport, you will still take part of these 2 and take it down here. Then for you to do that, you have to apply force. To apply force, you need energy. So it means when defining active transport, you must remember to say is the movement of substances from area of lower concentration to high concentration by the use of energy. Energy must be in the definition. So what are the food molecules that needs to be absorbed that way? Examples are glucose and amino acids, amongst others. I only gave 2 examples. I actually meant to mention one. I should just say more than that. It says here, why is it necessary to absorb some food molecules by active transport? You remember that this guy called glucose is what keeps us alive in terms of providing energy for us. Now, let me show you what I'm trying to say here so you're going to understand this. What I'm drawing on the screen is I'm trying to draw a blood vessel. That's a blood vessel there. Now, just follow me. This is a cell. Now, you have 1, 2, 3, 4, 5, 6 molecules of glucose in the blood, for example, please. And you have 1, 2, 3, 4, 5, 6, 7, 8, 9, and 10 in the cell. According to diffusion, substances will normally move from the cell like this. But we don't want that because we need glucose in the cell. So instead of using diffusion, the body uses active transport like that, saying that even though you have 6 molecules in the blood and you have 10 in the cell, the active transport, which is what came from evolution demands that you still take in from where you have fewer ones to where you have where you have more molecules. And why is that? Because we need glucose to be available at all times. All right. So why is it necessary? You could just say that to maintain availability of essential nutrients. Essential nutrients. Yeah, that's because it must be available. If it was going to be moved by diffusion, that means when we have more molecules in the cell to just move into the blood, and that is wrong. What we want is, even you have fewer in the blood than is this in the cell, you should still add to the one in the cell. That's what. So it is to maintain availability of essential nutrients or food molecules as you have it. So just why epithelial cells have many mitochondria. Well, if you've been following all along, we've talked about active transport. We talked about moving things against concentration gradient. So let me ask this question now. If you can define active transport, try and do that now. I guess you said it's the movement of molecules or substances from area of lower concentration to higher concentration by the use of energy. So the question here is, what's the function of mitochondria? So the first I'm going to say is that mitochondria produce energy. Yeah, that's the function of mitochondria. This helps to actively move substances into the epithelial cells. All right, so the point is, so it's needed to do that, so to say. So it provides energy. So for that reason, that's mitochondria. So for that reason, mitochondria helps to bring in, provides the energy that is needed for the substances to move in, so to say. All right. Now, some plants also carry out, it's not me, they do carry out active transport. Give one substance that plants absorb by active transport. You can make mention of just those nutrients in the soil: nitrate, magnesium. Sorry, let me write it in full. Magnesium, potassium. Sorry, I'm used to writing potassium. You can go on and on. But please, glucose cannot be in here because glucose is formed on the leaves during photosynthesis. So what they absorb is not the way we absorb glucose actively. They don't do that because theirs is formed on the leaf. However, of course, in the body of plants, you still have a place where glucose in the body, where it is being transported actively in the phloem and all of that. All right, let's move on. Now, it says here, the image below shows some muscle cells from the wall of the stomach as seen through a light microscope. All right, it says describe the function of the muscle cells in the wall of the stomach. All right. So here, this is 2 marks, 2 points. So I'm going to start by saying that muscle cells contract and relaxes. All right. This helps to mix the content of the stomach with the gastric, let me write that clearly, please, with gastric juice by a process called churning. Now, this is very important. You expected to talk about the function of the muscles and how does that work in respect to the stomach. It works by what? Contracting, relaxing, like that. So this is what it does. If you've seen my lecture, if I've taken you an organization, you'll have seen where I showed you an animation where the stomach was like this and it just moves like that. So what does it do? That's called churning. So when you eat the food goes in there and mixes the food with the stomach mechanically mixes the food with the gastric juice, which is made up of pepsin, hydrochloric acid, and what have you in the stomach. So sometimes when you hear some sounds in the abdomen, some of them might be churning, but it's not all the sounds here in your abdomen that means it's the stomach that is churning the food. No, but one of them could be really. It takes a professional to know which one it is, which we can talk about that now. All right, so that's the two answers there for the 2 marks. It says here, figure above is highly magnified. The scale bar is in figure above represents 0.1 millimeter. Okay. Use a ruler to measure the length of the scale bar and calculate the magnification in the... Okay, I think I would need... Okay, I have my ruler here. So I'm going to take your ruler like that. You're going to measure this out. So I'm doing that right away here. And I have... this is 2 points. Actually, this is going to be a bit relative because if I zoom this in, it's going to be larger. So let me just... if this was to be a paper, it's going to be the same, but because I'm doing on-screen recording, so what? Let me just make it not too wide. So this is what I measured on the screen is wow, this is 2.9 centimeter. Now, so let's do it this way. So what I measured from here to here from my ruler is 2.9 centimeter. Now, excuse me. So let's work things out. So the magnification is image size over actual size. Now, in this instance, actual size is 0.1 millimeter. Then the image size, which I just measured using that ruler, is the image size, 2.9 centimeter. Now, you can't work with it this way because this is centimeter and this is millimeter. It doesn't work that way. The unit has to be the same. So 1 centimeter is 10 millimeter. So it's going to be 2.9 times 10 millimeter over 0.1 millimeter. So sorry, let me punch my calculator here. So we have so 2.9 times 10, that's 29 millimeter. Sometimes I do this so that you don't just guess, please. It's going to be 29 millimeter divided by 0.1 millimeter. So the millimeter cancels each other out. So 29 divided by 0 divided by 0.9, that's 290. So the magnification is 290 times. So that means this muscle cell has been magnified 290 times. That's the answer there. Hope you understand. Please feel free to rewind the video to get that right. Unit matters a lot when you're doing calculations or questions on magnifications. Now, the muscle cell in figure above contain many mitochondria. What is the function of mitochondria? How many marks do we have? 2 marks. Okay, now when you have 2 marks, you know that's 2 points. Remember the first point has to do with the function of the mitochondria. Then you now talk about how that function relates to that diagram. Okay, so we have, this is a muscle cell. Okay, now like we know, I guess you should be able to answer this now. Mitochondria produces energy. That's what it does. Okay, this energy is essential for the contraction and relaxation of the muscle. You might not have to say the exact way, but the point is you're meant to make mention of the function of the mitochondria. You produce energy. Okay, we can say via respiration. Then you then talk about this energy is needed for the muscle to contract and relax. For muscles to contract, you need energy. That's the truth. Now, the muscle cells also contain many ribosomes. The ribosome cannot be seen in the figure above. What is the function of ribosomes? Oh, okay. That's for protein synthesis. All right, I hope I got right that clearly. Yeah. It now says suggest why the ribosomes cannot be seen through light microscope. Yeah, because light microscope has limited resolution. Now, talking about resolution is the process, is the ability to distinguish two close points as being separate. Now, it appears as if it's one, but high resolution means it's going to, it shows details. Let me put it this way. Let me redefine now. You can just say resolution is defined as how a microscope gives detail of the specimen of the diagram, whatever it is. But really, you're supposed to define it as the ability of a microscope to distinguish two close points as being separate. Yes. Now, my hands, you see my hands, these two fingers are together. My index finger and my thumb is like as if it is one. Like you can just probably say it's one. But if you look at this under the light microscope, you still be able to see there's a space there. Yes, like it seems as if it's one. So because of high resolution, it's still going to show that these two guys are not together. So that's what the electron microscope has, which is why it can be able to show ribosomes, mitochondria, show the content of the nucleus, the content of the mitochondria, not just showing the mitochondria. It could actually show you that when we draw mitochondria, we draw it like this, like that. Excuse me, a few seconds. Let me redraw that nicely. Now it has this shelf in it, cristae. How do we know it has that? I didn't draw that very well. It's because we're able to see it using electron microscope. Of course, the electron microscope also has grades of based on their resolution and capacity to magnify. All right, so this question, please don't write magnification, because even if you magnify, you will still not be able to see anything. You still apply appear blur. But electron microscope, if you apply appear blur, it's going to show you the detail, like distinct it out, so to say.